This is Dr. Caroline Peterson. Thank you for joining me today for the class, Where Are My Keys? Preventing and Reversing Cognitive Decline Naturally. I am a chiropractic physician, mixed methodology researcher, and retired midwife. I've been in practice 30 years, and I've done hundreds of hours of postdoctoral training in functional medicine and functional neurology. I was a research postdoctoral fellow for the NIH at Oregon Health and Science University in the neurology department. And I sat in urogynecology with the urogynecologists. This class is based in large part on the work by Dr. Peterson over at UCLA. He has a fantastic program for treating Alzheimer's naturally. And so we're going to use a lot of his modeling and intervention strategies, and that's going to be complemented by the training that I've done with Dr. Karazian and Dr. Brock in functional neurology, the Carrick Institute in functional neurology. I've also studied with Dr. Karazian, taking his classes in functional medicine, as well as Dr. Berger's work with functional pediatric neurology and medicine. So this is a 13-week course, and today we're going over kind of like introduction. So we'll be describing neurology, development, decline, how the systems work, and kind of like broad strokes of what's going on. Next week, we'll go over how to interpret the diagnostic labs and what to do about it, and how that fits in with the model that we're using for cognitive decline. On week 3, we'll go over the pro-neurogenic lifestyle, diet, and nutrition, and fit your lab results and into that, the different models of phenotypes of cognitive decline will be fit into the pro-neurogenic lifestyle. In week 4, I'm hopeful that you'll have your lab results back so that we can use some examples from real life and go over what that means together. So you guys would all be interpreting somebody's volunteered lab results. In the following weeks, weeks 5 through 11, we'll just have online discussion. And this will be for like you guys saying, this is working for me, this isn't working for me, this is what's going on, what's happening with you. Just everybody kind of like keeping abreast of each other and giving each other help and support. And then by 3 months in, week 12, we should be able to rerun the labs and see some changes. So that's why this is a 13-week course, because I want to have a chance to do those labs again, and then to revisit them and to, based on those rerun labs, to make plans for the next steps. So today we're going to go over epidemiology, different terms and definitions, how the brain develops and degenerates, and the mirror process in that. We're going to go over names and functions of different parts of the brain and the neurotransmitters, and we'll go over the paradigms for cognitive decline and dementia. Today, the lecture is going to not have any questions during the lecture, but there will be questions at the end, so you can put your questions in the chat section of the Zoom. You can also, if you're watching the video later, you can also ask the questions in the discussion board forum. And just as a reminder, my role is as teacher, not as clinician. And so I won't be like acting as a clinician here. I'll be helping you reason through things so that you can become your own clinician. When the video is recorded and ready, I will upload it to Thinkific, and then you'll have access to that throughout the entire course. Also on Thinkific, you have access to the PDF list of lab tests that you can get your PCP to order. or a doctor from the actual lab can order it, or I can order it if you're in Oregon or Florida for a $50 processing fee. On the dashboard, you'll also see the PDF of the PowerPoint and also the pre-class self-assessment. That's really nice to do beforehand so that you can kind of identify what systems may be a little bit under functioning in yourself. So one of the issues with cognitive decline is that most people accept it as normal. Biomedicine accepts it as normal. But what is biomedicine? Biomedicine used to be called allopathic medicine, used to be called Western medicine. It's the kind of medicine that's practiced by your MD, most DOs, most nurses. It's the kind of medicine that you receive when you go to the hospital, basically. That's called biomedicine. Of course, MDs, DOs, nurse practitioners can practice functional medicine or functional neurology, but they have to get additional training for that. That's not part of their curriculum. So in giving a class like this on remediating cognitive decline, the reason that this class is not being taken by every single person in the world is because of the normalization of cognitive decline. So that makes a huge barrier for change, because people say, oh, I'm having a senior moment. Oh, I have the normal amount of pregnancy brain. Oh, that's just a mommy brain going on for you. These are not, we associate cognitive decline with our age or with a special circumstance or situation in our life. These are not things that should be normalized. Cognitive decline at any age and at any stage in life is death of neurons and synapses, and that's not a good thing. So the sooner that we can understand that cognitive decline is not normal at any age or circumstance, and that there's something that we can do about it, the better off we'll all be, and the earlier we can intervene. So for this class, it's easy to feel overwhelmed by the materials. This class is designed at a graduate or postdoctoral level. So the things in this class are complex. The information is new, and I know that you are intelligent people and that you can master it. And at the same time, you don't have to know it all. You don't have to memorize anything. You can come back to anything that you want. You can take breaks. You can basically do what you want. This is for you, and you can study it to the depth that's right for you. Just remember that there's going to be lots of repetition over these 13 weeks, and that it feels like a lot because it is a lot. It's a lot of information to take in, and you can do it in a way that's right for you. That's totally fine. What I want to start with is the idea that there's hope. So many of us have seen loved ones decline. So many of us have loved ones in memory care units, and many of us have people in our family who have died of some sort of dementia, and we're afraid. Maybe I'm next. Maybe it's going to happen to me too. I see so many people around me experiencing this, and I don't want it, but maybe I'm predestined to have it. And I want to tell you that there's hope. When I was 20, I went to Spain to work as a nanny or au pair. And I lived on a street called Ramón y Cajal. And I didn't know who that was, Ramón y Cajal. I did not know I was living on the street that was the street of the famous Spanish neuroscientist who had won a Nobel Prize. I didn't know that. And I didn't know that I was living on the street of the person who has had a tremendous impact on the field of neurology through the 20th century. So in 1913, Ramón y Cajal said, nerve paths are fixed in the adult, everything dies, Nothing's regenerated. That's that. And so we all said, okay, sure. That was accepted. That was just, the way things are. And so we accepted it. In the 60s, people started doing more research on animals and found out, wait a minute, maybe that's not true. We see some neuronal regeneration in rats and cats. And then as time went on in the 2000s, there came to be many papers on human neuronal regeneration until finally by 2020, it's very well accepted that there is adult neurogenesis. It happens. We don't just fall apart. We can remake ourselves, and it happens in the brain. That's where it happens. And it takes, for a nerve to regenerate, it takes 4 to 6 weeks to make a new nerve. It can happen. We are designed to renew. And the place that it happens is right here. Right here. It happens at the bottom part of the lateral ventricle and that part of the hippocampus. This is where neurons are born, even as adults, even as adults. So Dr. Brednison's work out of UCLA on treating Alzheimer's came up with 3 different phenotypes for cognitive decline. That's going to be a mainstay in our modeling here. We're going to harken back to that over and over again. So these 3 phenotypes are inflammatory. These are the things that cause cognitive decline or Alzheimer's eventually. Inflammatory phenotype, the atrophic phenotype, and the toxic phenotype. So most people who experience cognitive decline and Alzheimer's will be part of this inflammatory phenotype. They're too inflamed. And so they experience cognitive decline because they're too inflamed. Some people will experience cognitive decline because they don't have the nutrients, the hormones there to be able to nourish the cells. And so their brains become atrophic. They're not well nourished. And so they experience cognitive decline because of that. Some people are going to experience cognitive decline because of exposure to toxins that have not been treated. And so whether these are microorganisms or whether they're heavy metals, environmental toxins, things like that, those are things that that need to be addressed in order to reverse cognitive decline. So as we're working with these 3 phenotypes, we're also going to be looking at the underlying structures and systems that contribute to those phenotypes. So we know that in order to have good cognition, in order for our brain to work properly, we have to methylate properly. We need to produce and transmit neurotransmitters properly. We have to have low inflammation, few infections. We have to have proper vascular flow and the ability to produce and use energy. And our barrier systems need to be working in our body. And we're going to go over all of this, so don't worry. This is just an introduction. In natural medicine or functional medicine, our goal, first off, is to identify if the blood-brain barrier is sealed or not. If it's not sealed, that's the first thing we got to do. We got to seal it up. After that, we're going to identify what systems are involved in your particular cognitive decline, which phenotype you fall under. And then we're going to, using the lab work, we're going to identify what factors need to be remediated. We're going to learn how to remediate those. And then we're going to go into the elements of the pro-neurogenic lifestyle, the lifestyle that we should all be living for the rest of our lives. And in that way, combining all of these things, we're going to rebuild neurons and synapses. So the epidemiology. Alzheimer's dementia accounts for most of the dementia by and large that we experience in North America and Europe. And it's twice as common in women as men. The second runner-up is vascular dementia. Then trailing, we have the frontotemporal dementia, the Lewy body dementia. And then outside of the, the disease states, we'll see mild cognitive impairment. And that means that whatever is going on for you has started to show up on testing. Before that, there'll be subjective cognitive impairment. That means that something's going on in your brain that you or a loved one notices, but by and large, people accept it as normal. That's where we want to enter in. It's ideal to enter in when it's just at a subjective stage before we have a diagnosis, because then it's much easier to reverse. So many of us are afraid of Alzheimer's disease because we know that there's a component of inheritance to it. There's a genetic component to it. So only 5% of Alzheimer's disease is the early onset. That's called the familial Alzheimer's disease. That's when you get it before you're 65. That's the one that goes so fast, like they make movies on that. But even with this early onset familial Alzheimer's, only 1% of it is related to genes, genetic mutations. By and large, most Alzheimer's disease is a late onset after 65. It's a slower progression. There are still genes that may impact it, but genes have to be turned on. Genes are turned on by epigenetics. What is epigenetics? Epigenetics is what you do, what you think, how you move, what you eat, who you love, what are your relationships like. That's epigenetics. That's what turns on and off genes. You have a lot of power. You have a lot of control in your life because you are in control of epigenetics and gene expression. We know the risk factors for Alzheimer's disease are, of course, advancing in age and being a female. There are also, physiologic conditions that put you at risk, which would be obesity, diabetes, cardiovascular disease, and then things you do that put you at risk, like smoking or physical inactivity. Alzheimer's disease has been researched for decades now with very, very little positive outcome, very, very little furtherance of our understanding of the disease. So in the early years, the clinical trials reaped no benefit from targeting the amyloid beta plaques and did create adverse outcomes. Then in the 2012 to 2017, again, we see no clinical trial that produced significant cognitive benefit. And yet we have drugs that are approved for treating Alzheimer's. And these drugs in the clinical trials they were part of showed no disease modifying effect. During the pandemic in 2021, we got a new FDA approved drug for Alzheimer's disease. It was fast-tracked. It was approved even though all the studies of this drug were terminated early because of the futility of the intervention. And also, those studies found that the intervention, although it didn't help Alzheimer's, it actually increased the risk for cerebral edema and intracerebral hemorrhage. Why did they approve it? So the argument for approving these drugs is that they give hope, and people need hope. And I'm going to tell you something. People do need hope. And we have hope here through the work that Peterson has done and through the protocol that we're going to be talking about with functional medicine and functional neurology. This is where the real hope lies without the drawbacks of the drug interventions. So what is wrong with all of these studies that are not figuring out how to fix Alzheimer's? So when you see something like this, what you're really seeing is that one of two things, generally speaking. One, the researchers are asking the wrong question. If you don't ask the right question, you're not going to get the right answer. If you keep asking the wrong question over and over again, it doesn't mean you get the right answer. You're still going to get the wrong answer because you're not going to get the answer because you're asking the wrong question. The other possibility is that they were just intervening too late, because in our culture, within the biomedical model, we don't recognize cognitive decline until the very end stages of it. And by the time you're in the end stages of anything, it's very hard to reverse it. And so in the functional medicine, functional neurology approach, what we want to do is to intervene early and have the greatest likelihood of having a positive outcome. So in 2016, 8.5% of the US population had dementia, population over 65. And this was actually down. It was down 30% from 2000. So that's awesome, right? It had gone down. Still sounds like a lot, but it was going down. And why? Why was it going down? So the number one reason for that prevalence going down was that people were becoming more educated. And this had a greater impact on men than women. Both men and women it impacted, but men more so than women. And this is saying, you need to exercise the brain. You have to learn. You have to think. You have to keep learning if you want to keep your brain healthy. And you can do that. That's epigenetics. Awesome. You can do that. It has a huge impact. What we also found out was that things you can't control are less important than things you can control. You can't control your age, your race, your ethnicity, but you can control your cardiovascular risk and your education. That's epigenetics. You have a lot of power. You have a lot of power in determining the outcome of your brain. So we came up to 2016. We were going down in the prevalence of dementia. And then we had COVID. And then we had long COVID. So in 2019, before COVID, Alzheimer's was the 6th leading cause of death in the United States. Between 2019 and 2020, with COVID, the deaths from Alzheimer's increased 145%. That's a lot. So we don't really know why. We don't know what goes into it. We don't know, well, was it because of COVID? Was it because of social isolation? Was it because of surveillance, more or less surveillance? We don't know exactly what all feeds into that. And so we're going to withhold judgment, because when we're talking about data, it takes time to clean it. It takes time to come to the big picture. And so we're just going to say, this is what we're seeing now. We don't know what we're going to see in a few years as the data is cleaned up and we understand it better. This is what we're seeing now. Another thing that is coming out in the literature is that people who were 65 and older and had COVID were at 69% more likely to get a new diagnosis of Alzheimer's within the next year than people who had not had COVID. So again, we're going to take this information and we're going to say, that's interesting. And let's also watch and see what happens as more data is crunched and as we get cleaner data. So long COVID is a thing. So long COVID now does not have a uniform definition. It's a definition that's changed over time and is still perhaps changing regionally. And so perhaps in part because of this and because of the diagnostics, then it's unclear exactly what the data say. So when we see a large variance in data reporting. So we're seeing 5 to 45% of people who had COVID will experience long COVID. Like that's too big. That's too big of a range to be meaningful. So something's wrong with the data. And so that has to be cleaned. But even if it's only 5% of COVID, people had COVID who get long COVID, it's still a lot of people because a lot of people had COVID. And so we're withholding judgment. We're waiting for better data. We're keeping this in mind. Long COVID's a real thing. And we also know long COVID has a lot of cognitive impacts. So again, because the data is early, we're not seeing like very systematic use of time and dates and diagnostic criteria, things like that. It's messy, still messy data, needs to be cleaned, but we do know that cognitive decline consistently is a component of long COVID. And it's not surprising because long COVID is a post-viral syndrome. And post-viral syndromes are not new. People suffer post-viral syndromes day and night. And they are very happy that now more money is being thrown at post-viral syndromes, long COVID, because all the post-viral syndromes are going to benefit because we're going to understand them better and we're going to know better what to do to intervene and what's going on. So for this course, the intervention model that we're using is drawn from functional medicine and functional neurology. And so that model is all about the ideal. It's about approximating the ideal. So as soon as something starts moving away from the ideal, we want to identify it and bring it back. Early intervention. So, and natural intervention. When things happen like that, then it's easier to make intervention and natural intervention. So with biomedicine, this is not the way that things go. So there's a, here in this graph, we're seeing functional capacity, degeneration over time. And this is awesome. This is your functional capacity, the way that it should be. And in natural medicine, in functional medicine, in public health, we want prevention. We want prevention so that we don't even have to go down this road. And so that's the first thing, prevent pathogenesis. That's what we try to do. If that doesn't work, if there's an exposure, if we have initial damage, then what we want to do is address that. Here we go, right here. Look at that. You're still pretty highly functional. Awesome. Let's take care of it. See if we can bring you back. If that's not intervened there, secondary damage develops, and then we're like, okay, come on, let's do it. Got to do it now. Intervene now. Try to bring us back up to a higher level of functioning. If we don't do it with a secondary damage, then finally there's some pathology that's beginning to impact the physiology. We're getting synaptic loss. There's more inflammation. And this is like the time that biomedicine is starting to think it might be a good time to intervene, but a lot of times we just want to wait until there's the clinical syndrome, which is the diagnosis. So a lot of times biomedicine is going to wait until you get your diagnosis. At this time, it's very difficult to reverse things. You're far down this slippery slope, and many times you're just into symptom management. Two very different models. So in the biomedical model of cognitive decline, look, everything's awesome. You have normal cognitive function. That's stage 1, is normal. Stage 2, you can't recall your loved one's name. Everything's normal until you can't recall your loved one's name. So in functional medicine, that's not the way we see it. We want to intervene way before you can't recall your loved one's name. We want to intervene when you're like, now, why did I come into this room? Wait, why did I start this sentence? I know there was a point. Things like that. That's when you want to intervene. That's the early stage. That's when you want to intervene. That's easier. So this reminds me, this model This biomedical model of cognitive decline reminds me a lot of my nephew's marching band grading. So all the marching bands in the state compete. They have something, let's say like a 300-point scale, 300 points. And then the top, let's say 3 bands, 5 bands, something like that. They're going to be at the very top of that 300-point scale, and they're going to be within like 1 point or half a point from each other. This is not a good scale because it doesn't parse people out enough. And this is not a good scale. This biomedical model is not a good scale because there's too much normal and then a precipitous drop. And so this is not meaningful. This is not, to me, it's not helpful. That's why I like the functional medicine approach better, because it's better gradated so that you can know, identify something is wrong before you can't remember your loved one's name. Something is happening before then. So in order to understand better how the brain works, how the brain is supposed to work, it's helpful to look at how the brain develops, and then to also compare that to how the brain declines, because these are like mirrors of each other. And that's what we're going to use to help understand. So newborn babies are spinal cord and brainstem based. They do not have cortical function. They have no voluntary nervous system. They're not making decisions. They're behaving reflexively using the primitive reflexes, the brainstem, and the spinal cord. When babies are born, they're born into flexion. That's why we say fetal position. Fetal position, everybody knows, oh, that's flexion, right? That's the way babies are born. They're born into flexion. And they're born in sympathetic dominance. So that they're like a little bird. Their heart is racing so fast. They're breathing fast, but also irregularly. And they get cold so easily. They have to be all bundled up because they have poor blood flow. That's all sympathetic dominance. They can't control their pee. They can't control their poo. They're wearing a diaper. When you feed your baby, you're like, is it going up? Is it coming down? I don't know if they're going to spit up. Is it going to be a blowout? What's happening? Is my baby pooping many times a day, 1 time a week? You know, sensitive digestive system. Did I eat something wrong? Now my baby is colicky, you know, and then that baby is mostly going to sleep, mostly sleep. And this is normal. This is a normal newborn baby. Normal newborn baby, spinal cord, brainstem based, sympathetic dominance. That's normal. So here's the brain, right? The cerebellum and the cerebrum. That's the brain. That's not really very active. And then here's the brainstem. We have the mesencephalon, the pons, the medulla oblongata, and the spinal cord going all the way down. So that's what's going on. Let's talk a little bit about terminology. So a neuron is a cell that transmits information, signals, right? So this is a neuron, and it's composed of the dendrite and the axon. So the axon is what sends the signals. The dendrites receive the signals. They hook up together, and where they hook, it's called a synapse. Synapse, that's where they hook up. And then the information goes between the synapse, and the longer it stays in the synapse, the more it excites the synapse. And if it's covered with a myelin sheath, which is fat, then the faster the information flows. So a nerve is made up of a bundle of neurons. A neuron is a cell. A nerve is a bundle of neurons, which is a bundle of cells. Now, when the baby is born with a bushy brain. What does that mean? Bushy brain. The baby is born with more neurons, more cells in their brain than adults. And during the first 1 or 2 years, their synapse density is 50% more than adults. And yet they're born without real activation of the brain. Why? Because they don't have synapses when they're born. It's the synapse that makes the information carried. They have a bunch of neurons, and it takes time for that communication system to be set up. So babies, when they're even in the womb, they're starting to produce myelin, but that myelin, their system is not well myelinated at birth. It takes time, it takes years for the nervous system to become myelinated, when it's myelinated, it goes faster, it's more efficient. And so that's what takes time. That's part of neurologic development. That takes time. And so in order for this bushy brain to become efficient and effective, what we need to have happen is pruning. It has too many neurons and too many synapses to be efficient and effective. And pruning is what helps it to function better over time. So when the baby is born with all those neurons and develops all those synapses, what is that? Why does that happen? That happens because Mother Nature is planning for all possibilities. Those neurons are latent possibility for that baby. That brain does not know what exposures that baby will have. That brain does not know what environment that baby is born into and what connections need to be made. That brain has a pre-existing building blocks for any environment so the baby can succeed. Latent possibility. But to be able to succeed, the brain has to be pruned so it has the right networks for that particular environment. How are those networks determined? How does the brain know what networks are needed? Well, what we say is sensory input, motor output. So this is the way the brain is built. The network is created by touching your baby, rocking your baby, singing to your baby, feeding your baby. This is sensory input. And then the baby first is reflex-based. We got the primitive reflexes. So the baby is responding based on the reflexes. And with enough response, enough sensory input and reflexive output, the voluntary nervous system begins to form. The voluntary nervous system begins to form. And the first thing we see is when babies are born, their thumbs are like this. And then in a few weeks, you see your baby go like this. Your baby's brain is developing. That is part of the cortical system starting to fire and determining position and movement in the body. So a lot of the primitive reflexes should be extinguished by 3 months. If the baby has enough sensory input, motor output, you got to use it to lose it. Use that primitive reflex to be able to integrate it into part of the voluntary nervous system. That's the way it goes. If you don't get enough sensory input and motor output, then what's going to happen is that you're going to have like all these neurons, all these synapses that are going to be like talking. And then you really have a very weak and jumbled system. And if you get too little pruning, that, in that kind of situation, you're going to get something like autism. If you get too much pruning, you're going to get something like schizophrenia. And so what we want is just the right amount of pruning. The right amount of pruning so that your baby can succeed in the environment in which they were born. Sensory input, motor output. So the experience that babies have, which I said, sensory input, motor output, is what determines the pruning. What wires together, fires together. Do it enough, and that's the network. If a baby does not have an experience, then they're not going to have that wiring. They're going to have dieback. And then, it's something it's a movement, it's a thought, it's an experience that's not necessary for that environment. And so the brain is efficient, and it'll be like, well, don't need that one. Let's work on the ones that we do have. Let's run those. Let's run that program. So this is the way the body's designed. And we can see it in imaging, so that if a child is neglected, their brain is not going to develop because they do not have the sensory input to build the motor output. They do not have the environmental demands that require that brain to develop and grow. That kind of looks like the Alzheimer's brain. If you look at the normal and the extreme neglect child brain, and you look at the normal and the Alzheimer's brain, there's a similarity. The Alzheimer's brain is smaller, just like the neglected child. The Alzheimer's brain, we have diminished white matter, gray matter, enlarged ventricles, and enlarged sulci. The mass is less, just like in that extreme neglected child. So what does a baby need for normal neurologic development? This is epigenetics. A baby needs mental and physical exercise, nutritionally dense food, social engagement, and avoidance of pathogens. When a baby has that input, they will go into normal neurologic development. Normal neurologic development is moving a baby from being spinal cord and brainstem based into being cortical based. That means having a choice. The voluntary nervous system is developed when a baby becomes, or a person becomes, cortical based. The baby, normal development for a baby is moving from sympathetic dominance to parasympathetic dominance. Normal neurologic development is moving from a brain, behavior, and thought that is less efficient into things that are more efficient. Moving from responses that are hypersensitive and intolerant into responses that are flexible and adaptable. Moving from that flexed fetal position into the upright standing position. So babies are sensitive to everything. They're sensitive to sound, to light, to movement. They're sensitive to temperature, all these things that they need a lot of cushioning so that they can have their needs supported. That's just part of their development, right? That's normal for a baby to need those things so that later they can become flexible and adaptable. So part of this is, moving from the sympathetic into the parasympathetic. So babies are born, we say, oh my gosh, that baby is just so beautiful. Look at those big eyes. And when we say, look at those big eyes, what we're saying is, look at those big pupils. Babies are born with very dilated pupils. And no wonder they're sensitive to light, right? If you have big, huge dilated pupils, then you're going to be sensitive to light too. And it's as this parasympathetic system develops, then there's pupillary constriction. So this is part of the autonomic, right? So autonomic nervous system is part of the nervous system that is automatic. It happens automatically. And you don't have to, it's not part of the voluntary nervous system. It's deep, it's hardwired. So also, when babies are born, they don't have a bunch of salivation. And so when people bring their babies in, they'll, 2-3 months, something like that, then they'll be like, oh, my baby's teething. Look, they're drooling. And I'll be like, oh, your baby's coming, this parasympathetic is getting turned on. That's awesome. You have to wait a few more months till they teeth. But yeah, so salivation, same thing. So salivary, inferior superior salivary nuclei are for saliva and tears. So then you see also these babies around the same time, they're going to start to have real tears when they cry. So it's not just going to be the sound, but they're going to have tears come out. And that's also development of the parasympathetics. As the baby develops, their heart rate is going to go down. They're going to have more stable breathing. They're not going to eat all the time because they can control their glucose release. They're going to be able to start controlling their bowel and bladder. And they're going to have a calmer, more predictable digestion and not have the colic. So as the babies develop, they'll move from being sympathetic dominant to parasympathetic dominant. Now, when you're parasympathetic dominant, ideally, you still have sympathetic tone. So you should be about 30% sympathetic, 70% parasympathetic. That's when you have the right tone in your body on kind of like a regular basis. This is like kind of like your baseline. And this will enable you to do all of the basic stuff of life, which is to have a baby, enjoy sex, enjoy your meals, poop after your meals, control your pee, all that kind of stuff. That's basic. And that's what happens when you are in correct balance between parasympathetic and sympathetic for a mature or adult nervous system. Now, part of what brings us into that mature system is something that's called inhibition. So the body naturally wants to revert to sympathetic dominance. And in order to prevent that from happening, we need to have inhibition. And that inhibition of sympathetics is vis-a-vis the vagus nerve, which is the 10th cranial nerve. We also have a tendency to have a lot of limbic distortion if we don't have inhibition by the prefrontal cortex. These are things that develop with time. So we have the vagus nerve and the prefrontal cortex that are the two main inhibitory systems of the body. We need to have these systems mature and working well in order to be pro-social, to be happy, and to be efficient in our bodies. This is the good life. The good life is the life of inhibition. When we don't have those inhibitory systems working, we're going to have problems with people. We're not going to feel good in our bodies. We're going to be kind of deranged. That's not what we want. So how does this happen? So what happens with babies is that the mesencephalic reticular formation here is going to excite the sympathetic response through the spinal cord. And this is just firing. This is a standard firing away. And so in order for that not to happen, what we have to do is bring in the inhibitory system. And we do that cerebellum. Cerebellum is about movement. Cerebellum is about helping us to move efficiently. That's going to come into the brainstem and also overflow into the cortex. And then the cortex is going to bring that information down into the vagal nuclei and the pontomedullary reticular formation. And it's that vagus nerve that's going to fire down into the spinal column and inhibit the sympathetic response. So that's how it works. So this is what we need in order to be parasympathetic dominant, in order to be pro-social. Now, the maturation of the autonomic nervous system does not occur in just the snap of a finger. It's not like that. It takes about 20 years for that to occur. And the prefrontal cortex takes even longer. That takes about 25 years. And if anybody's met a toddler or a teenager, they know this is true. Because these people are very impulsive and they're very, egocentric. They're not pro-social, or they're pro-social to an extent that's very picky about their social. So the question is, how do we get from being spinal cord based to cortical based, right? How do we come into neurologic maturity? How do we come from being sympathetic dominant to parasympathetic dominant, less efficient to more efficient, hypersensitive and intolerant to flexible and adaptable? How do we come from being flexed into bipedal, upright? How do we do that? What babies need for normal neurologic development, again, is physical and mental exercise. They need input, sensory input, nutritionally dense food, social engagement that's diverse, and avoidance of pathogens. That's what a baby needs. Again, if they don't get it, they're not gonna develop neurologically. And if we don't get that as adults, we're gonna have the same thing. We need that as grown-ups too, and we're gonna have cognitive decline, and we're gonna have that loss, that mirrored loss, if we don't get the things that the brain needs. So what is normal aging? Just like I said, normal aging requires mental and physical exercise, regular, nutritionally dense food, social engagement, avoidance of pathogens, or treating them. If this happens, if the brain, if the body gets what it needs, there is very little pruning and dying back of neurons and synapses. That is normal aging. We have to get that into our heads. That is normal. We don't have to experience this cognitive decline. This is normal. This is normal. Steady state, normal. It takes something. Do you think a baby is just laying in their bed and then all of a sudden gets up and walks and talks? No, that's not how it happens. Babies have to do all this stuff in order to become upright and develop neurologically. We do too. In order to maintain it, we have to do all that stuff to maintain it. That's just the way it is. We're in a body. Body needs certain stuff. Give it to the body, and the body's like, awesome, let me do my thing. So, big thing here, there's a conflation of terms. Aging does not mean brain degeneration. Normal aging has very little brain degeneration. But you got to do the right things to nourish and nurture the brain and the body in order to have normal aging and normal brain function. Now, abnormal aging is brain degeneration. I'm calling it. It's abnormal aging. Abnormal aging is brain degeneration. That is what we have accepted culturally as normal, and I'm saying it's not normal. Abnormal aging and brain degeneration, you go from being cortical based to spinal cord and brainstem based. You go from being parasympathetic dominant to sympathetic dominant. And you'll see this. Low parasympathetics, you're going to see the dry eyes, dry mouth, you can't swallow, and you can't poop. People are using all kinds of drops, getting all kinds of cavities because they don't have saliva, can't take their pills anymore, go down to a smaller pill or transdermal or something like that. They can't swallow. They have to put their head in a special position so they can swallow and not choke themselves. Can't poop anymore. Low parasympathetics. That's trade-off is high sympathetic. Who doesn't have hypertension? High heart rate. Have to wear a sweater. So cold. I need my socks and my slippers. And then blows up over little stressors. This is all high sympathetic. That is what happens when there's a shift in the autonomics and we go back in time to what a neonate is like. We're going back. We're becoming less efficient. We're becoming hypersensitive, intolerant. People bug us. We can't put up with them. Where any little thing fly off the handle. And our bodies change. We come into a internal rotation, stooped position, forward lean as we lose the firing of our extensors. Voluntary nervous system down regulates and we go into that flexed position. It's not good. That's abnormal aging, and that represents brain degeneration. Brain is getting smaller, losing mass and function. It's not able to inhibit anymore. That vagus nerve is slowing down. It can't fire as well into that spinal cord to suppress the sympathetic response. So we're going higher in sympathetics, lower parasympathetics. And we're seeing frontal escape. So, the frontal lobe, so see this whole thing here, this all is the frontal lobe. This here, this is a prefrontal cortex. This is the part we're talking about. This is the part that inhibits. This is the part that's not developed till you're 25. This is the part that dampens limbic activity, and that is prosocial. When you have frontal escape or disinhibition of the prefrontal cortex or downregulation of the brain in this region, you are getting crazy antisocial behavior. People who can't get along with each other, you get road rage, you get people who bark at each other, who blow up at each other, in the supermarket over nothing, people who are impulsive, people who can't wait their turn, and people who make terrible decisions. And this is just because their brain is degenerating. Now, this can be reversed, but you have to do something to reverse it. It's not going to reverse on its own because this is an add-on. This is an extra special piece of being a person that takes work. Now, what we're going to talk about, we're going to compare the models of cognitive decline, the functional versus biomedical paradigms. So in biomedicine, as we've been saying, normal aging is just normal cognitive decline. Oh yeah, you got the normal amount. Oh, that's a senior moment. Yeah. I got my pregnancy brain going on. Got my mommy brain going on. These are inflammatory states. These are not normal. There's not a normal age or situation for cognitive decline. These are not normal. These are things that should be avoided and reversed. But in biomedicine, there is an expected decline in memory and thinking with age. And so, like we were talking before, the first time you see that in biomedicine is when you show up on a test as mild cognitive impairment that will then develop into dementia. So this subjective cognitive impairment that you notice your memory's not as good, or your friend or family member is like, oh man, they're not quite as sharp as they used to be. On the test, everything's perfect. You pass with flying colors. I can draw a clock, I can identify a camel. I'm sorry, that's not a very good test. That's that you could pass that test with flying colors and still, in a functional medicine and functional neurology perspective, still have some impairment because we are setting a different standard. We're going towards ideal. So when we go towards ideal in functional medicine, functional neurology, we're really trying to prohibit the death of neurons and synapses. We don't want that. We want vibrancy. We want to stay at this high level of function and have very, very little decrement, very little, and bring it back when we can. In early intervention, early identification, bring it back. High level of function our whole life long. Biomedicine, again, everything's normal until you can't recall your loved ones' names. Okay, that's not enough. Sorry, that's not enough. You deserve more than that. So now think about this. The way the brain is designed, if you have any kind of weak area of function, that is going to be what goes down the drain first. So let's say you've never been strong in math. Maybe you're not good at spelling. Maybe it's hard for you to find your way around, to orient in space, that drive, stuff like that. This is going to be the thing that goes down the drain first, which means that should be the thing that you really work on first, because you want to get that stronger. You want to bring up the function of all the parts of the brain. So in functional medicine, functional neurology, the first signs that we see of brain degeneration are fatigue. Fatigue when you use your brain. Ooh, I can't read as long as I used to. Now I get tired if I read. Ooh, I can't drive as long as I used to. I have to take breaks. Oh, that conversation, I kind of get lost in the conversation, or that conversation is boring. I'd rather just listen than enter into it. And the hippocampus and the neurotransmitter acetylcholine are the first involved with brain degeneration of this type. So you'll see the right hippocampus is what goes first, and the right hippocampus is where we orient in space and location. And that's where we're like, jeepers, where did I put my keys? I lost those keys again. Where are my glasses? Oh, they're right here on my head. Where did I park my car? You know, that kind of thing. That's right hippocampus. That goes first. Those are the kinds of things that we're like, hey, oh, there they are. That's so funny. Oh, you lost your keys again. Can't you just put them down one place? No. Then the other one, the left hippocampus, the other side, is like, oh man, it's on the tip of my tongue. Like, what's that word? Well, I'm just not going to think about it. Then it's going to come to me. That's also loss of brain function. And then you start seeing affective disorders. So you start seeing, man, I kind of just feel blue. Oh man, that makes me more nervous than it used to. So, I mean, who doesn't have a affective disorder nowadays? Everybody's depressed, everybody's anxious. These are all signs of brain down functioning. It's something that we've come to accept as normal, as natural, as just a part of who we are. The other thing is that is an early sign is poor digestion. So whether that's diarrhea, irregular stools, constipation, GERD, if it's oh, my food is so heavy, you know, things like that. Those are all early signs that the brain is not functioning up to par. And of course, in the biomedical model, you're still normal. You can still remember your loved ones' names. Now, the next stage in functional neurology, functional medicine, is the intermediary stage of brain degeneration. And that's when you have a shorter attention span. Oh, I only like to watch videos that are, you know, several minutes long because then I get bored. I get bored. Well, you're getting bored because you don't have an attention span because your brain is fatigued. You can't pay attention longer because your brain is fatigued. I'm not saying this in a mean way. I'm just saying from a functional medicine, functional neurology perspective, that's why you have a short attention span, because you don't have the fuel. The fuel is not there to help you maintain a longer attention span. And this is where you see people who are like, man, in my day, in the olden days, when I was a kid, when I was young, these were so much better. And this is terrible the way things are now. And well, I don't want to learn that new technology because what's the point? And it was better this other way. And I don't really trust this and that new thing. And it should be back the way it was. So these folks are kind of like stuck in the mud. They have difficulty learning new things, but they're not going to acknowledge that. They're just going to like harken back to the good old days, and then not have to learn new things, because in the olden days, everything was better. By the time you get over here to this intermediary stage, you're going to have chronic constipation. These folks can't go to the bathroom. And that's going to be, you know, that's the decrease in parasympathetic. And then we got the increased sympathetic, which is the high blood pressure, the high resting pulse. Everybody's cold, losing their hair, can't have sex. And then everything just throws you off. Everything's super stressful. And so you just are paring back your life, making it smaller and smaller so that it's tolerable for you. And in the biomedical model, you're normal. You can still remember your loved one's names, so you're fine. Now, when we get to the advanced stages of brain degeneration in functional medicine, functional neurology, this is when you see people are like, I think it's time for me to retire. I'd rather do something else. Like, they're not going to be like, my brain is degenerating and I can't focus or work as long. I don't have the energy. They're going to be just like, I deserve to go on vacation and I've worked a long life. And, so there's a conflation again, like happens about the time people retire. And so they're not like, oh, it's brain degeneration. They're like, oh, it's time for me to retire. I want to do something else that's less demanding. And at the same time, you'll see like people generally part of the affective disorders is that there'll be less expressiveness in the face, and then you'll get more grumpiness, people complaining, and a lot of that going on. And still these folks are like, oh no, everything's fine. They're not going to be aware of any neurologic loss, and maybe somebody in the family or maybe someone else who cares about them may be like, this person is experiencing some change, and they'll be like, pish posh, everything's fine. I didn't want to do that anyway. And in the biomedical model, everything's normal. Awesome. You still know people's names. So finally, when we get to severe brain degeneration in the functional medicine model, this is where we meet up with the biomedical model. This is where we have a name. We have a neurodegenerative disease. We have bladder incontinence, if not before, by now, certainly. The constipation has developed to such a degree that we have bowel obstruction, lost taste and smell, and the inability to develop social relationships. People just are like pulled back into themselves. And this is where people start to really rely on medical staff and family for meeting their daily needs. This is a diagnosis, and this is biomedically appreciated brain degeneration. And about the time that you may stop remembering somebody's name. It takes a long time to get to stage 2 in the biomedical model of cognitive decline. A lot of water under the bridge by the time you get there. A lot of things that functional medicine is going to want to address and stop and reverse. Functional medicine, right here. As soon as you come, we're going to be on it and reversing that thing as best we can. We're not going to wait. We're not going to have like, oh yeah, let's have a little watchful waiting here, maybe later. Okay, now we can diagnose you and now we'll treat you and manage your symptoms. That's not what we do in functional medicine. Early intervention, right back here. Prevent and then intervene early, right there. Keep this high function all our lives. That's what we want. I'm going to take a little break here. We'll come back. Going to pause. Why does cognitive decline occur? So in the functional medicine model, adults need mental, physical exercise, nutritionally dense food, social engagement, avoidance of pathogen for normal neurologic retention. If we don't have those things, that's going to start leading us down the slippery slope of cognitive decline. We know that certain things increase the risk for dementia. After a stroke, you have 2 times the risk of vascular dementia. After a moderate or severe traumatic brain injury, you have 2 to 4 times increased risk for dementia. What's not mentioned is mild traumatic brain injury, and how if you have one mild traumatic brain injury, maybe it's no big deal, but what happens is that the microglia, which are the cells inside of the brain that are responsible for cleaning up things after trauma, they get kind of primed. And so with the second, even mild traumatic brain injury, they start becoming inflammatory. And with the third traumatic brain injury, even mild, those microglia are not going to turn off. And so you're going to have an inflammatory state that's ongoing in the brain. And what we also know is that you don't just get a traumatic brain injury or concussion by hitting your head. You can also have a traumatic brain injury by something that's called a chemical concussion. So it can be even like eating an inflammatory food or having exposure to a mycotoxin or to heavy metal, or many different things can create a chemical concussion, which will provoke overactivation of the microglia and create that highly inflammatory state in the brain, which is, of course, a risk for dementia. We know now that oral hygiene has huge implications for the brain. The heart has been known for a long time. Now we know the brain. Well, why? Anything that happens in the face, the mouth, the throat, the sinuses, the nose, all this stuff is directly connected to the brain. And so very easy for any kind of a pathogen, microorganism to get into the brain from there. Diet increases the risk for dementia. If you have a poor diet, if you have GI problems, if you're not eliminating properly, if you're using medications like proton pump inhibitors or TUMS that make it so that you can't digest your food, then that's bad for dementia. You have to poop, you have to pee, you have to be hydrated, you have to sweat, you have to stay away from the pathogens. And even a lot of the medications are gonna like slow down and kill the mitochondria. And so those are all gonna be pro-dementia. Going back to our model. Our model is that there are 3 phenotypes for cognitive decline. Inflammatory, by and large, the most common. Atrophic, there's not enough nutrition, hormones. Then the toxic, those are what we're building on. Reasons for cognitive decline. So how does it happen? What are the mechanisms of pathology for neurodegeneration? This is what we need. We have to have a strong barrier system. We have to be able to methylate. We have to be able to produce and transmit neurotransmitters, low inflammation, good vascular flow, ability to produce and use energy. That's what we need. Those are the things that we need. Now, talking about the barrier system, everybody knows about leaky gut. You can also have leaky brain, leaky brain, leaky brain. So the brain also has a barrier, the blood-brain barrier. This is a semi-permeable membrane that keeps the bad stuff out. And if it's sealed, then you're largely safe. You're largely protected. If it's breached, then you're at risk. So it can be breached by an injury, disease, inflammation, any kind of free radical formation, and then calcium, all the calcium mediated injuries and problems are highly inflammatory. And so they're gonna also create a breach in the blood-brain barrier. Any kind of exposure to pathogens will have a likelihood of doing it. So what happens when there's a breach is that you get this leaky barrier system. And so stuff can get in there. You don't have the same signaling, neurologic signaling, you get dysregulation, you get neurodegeneration, neuroinflammation, and then ultimately you get death, cell death. And so we have to reverse all of that, especially before we treat anything like heavy metal poisoning. You do not want to have a leaky brain and be chelating out heavy metals, because your brain is going to be like sucking them up. So before you do anything, you have to check for a leaky brain. You have to seal it up. And how do we check for the leaky brain? We check for the leaky brain with GABA, the neurotransmitter. So in order to check for a leaky brain, a leaky blood-brain barrier, what you do is you, on a day at a time when you don't have a bunch of stuff going on, you have about an hour of free time, you're not too tired, you feel fine, you'll take a gram of GABA, and then you'll see, how do I feel for the next hour or two? Notice how you feel. You could feel like nothing. You could feel sleepy or relaxed. You could feel energized. However you feel, just notate it. And then next week, we'll talk about it. So try to do this week. You can get it at the health food store. It's pretty cheap. 1 gram of GABA. So what we know for sure is that neurodegeneration happens with Alzheimer's disease decades before you have a diagnosis. Decades. And that in functional medicine is when we want to intervene. We want to intervene when you're stage 1, according to the biomedical model. Everything's normal. You can still remember people's names. That's when we want to intervene. So what is neurodegeneration? So neurodegeneration is mitochondrial dysfunction coupled with protein misfolding. What are the mitochondria? The mitochondria are the powerhouse in the cell. They create energy, and the energy they produce is ATP, and that's what the body runs on. They help with the immune system. Again, calcium, you're going to see calcium all over the place. Calcium homeostasis and apoptosis, which is programmed cell death. This is a way the body cleans up after itself. Mitochondria are even related to stem cell regulation. And then, of course, we do have mitochondrial DNA, and that's the maternal DNA that's passed down through the female line. So here we have the mitochondria that's producing ATP. This is with oxygen. What damages mitochondria? A bunch of drugs damage mitochondria. Alcohol damages mitochondria. Over-the-counter pain medication. And of course, they can also be damaged by the apolipoprotein E4. And that's the one we'll talk about next week. Is related to Alzheimer's. So if we have problems with the mitochondria, like uncoupling, then the problem will be that it's unable to make ATP. So it can't make energy. That's a problem. We need, I guess I can't move that. Oops. We need that mitochondria to be making energy. It's the free radicals and the oxidative stress that cause the uncoupling of the mitochondria. Remember that free radicals, that's an unstable molecule, be an atom, or a molecule. It's that it's missing an electron. The body doesn't like uneven numbers of electrons. It wants to have an even number. And so then it's like, let me get another electron. I need an even number, and it's looking for it. If you don't have an antioxidant, then it's like, I'm going to grab it from here. And then that's going to cause other problems. So oxidative stress is when free radical steals an electron from another atom in the presence of oxygen. That's all it means. It just is not good. So another problem with the mitochondria is that if it can't block the NMDA receptor, then inflammation is created. So the uncoupled mitochondria can't block it. The coupled mitochondria can block it. And if the NMDA receptor is unblocked, then again, here's calcium. Calcium is highly inflammatory. And when the calcium rushes in, it's supposed to be counterposed by magnesium, but without the mitochondria there, it's not counterposed. And so then we get neuronal death, and that leads to inflammation. Remember that the NMDA receptor is a glutamate receptor. Glutamate is excitatory. It's an excitatory neurotransmitter. And so you're getting this excitatory neurotransmitter that doesn't have a place to rest. And so it's also like it's seeking, in a seeking mode. The third problem with mitochondria is that when you have the destruction of the mitochondria, because there's no energy in the neuron is going to die. And so that's not good. You need your neuron. So we talked about neurodegeneration being a combination of mitochondrial dysfunction and protein misfolding. Now we're going to talk about protein misfolding. So proteins are made out of amino acids. Amino acids are the building blocks of proteins. Everybody takes amino acids in their smoothie, something like that. Amino acids are like a chain, and then they kind of hook up, and then they kind of fold up, and then they kind of bunch up, and that's what is a protein. Protein is a bunch of different amino acids combined. So they have to fold in a certain way in order to be able to receive things and transport them or do the job that they're designed to do. They need to be folded in a certain way. If the protein is misfolded, then you're going to end up with disease. So like Alzheimer's disease has the misfolding of the amyloid beta and the tau protein. And that's what we're going to be kind of focusing on for this. So this is the amyloid beta plaque. Amyloid beta plaque is made out of a bunch of different misfolded amyloid beta proteins. And then the tau neurofibrillary tangles is also made out of misfolded proteins. We're going to talk about amyloid beta plaques first. These amyloid beta, the protein, is produced by astrocytes in the brain, neurons, skin, skeletal muscles, intestinal epithelium, the cells that line the intestine, and even bacteria. Bacteria produce amyloid beta as a biofilm. I didn't know that. And it has a normal function. Normal function is to protect from infections. It's as good as drugs for antimicrobial effects against 11 different identified microbes. It suppresses tumor growth. It can repair the blood-brain barrier. It promotes recovery from brain injury, hypoxia, and even autoimmune disease. It regulates the glutamatergic and the cholinergic synaptic function. So remember how we were just talking about the NMDA receptor and how that's glutamatergic? Everything's going to start fitting together. It also contributes to memory consolidation. It's a great protein, amyloid beta. So what's supposed to happen? It's supposed to do its job, and then you're supposed to poop and pee it out or sweat it out. Sounds pretty good. But if it forms plaques instead, then what's going to happen is, again, the protein is going to be disrupted. We're not going to have, the calcium. We're not going to have homeostasis anymore. That leads to inflammation, dysregulation of energy and glucose metabolism, mitochondrial oxidative stress leading to death, and then neuronal apoptosis, which also is death. Amyloid plaques do. But what causes them? This is a dominant model. What causes them to form? So there's something that's called the amyloid precursor protein, APP, amyloid precursor protein. It's a normal protein. It crosses the nerve cell membrane. That's what it's supposed to do. And it's supposed to be pruned by this enzyme called alpha secretase. Top part cut by alpha secretase, bottom part cut by gamma secretase. And no plaques form when it's done properly. It should be a peptide chain that's 38 peptides long. The body's like, awesome, I know what to do with that. Send it off to do its job. But sometimes what'll happen is that instead of alpha secretase doing the cutting, you'll get beta coming in. Beta secretase will come in. And sometimes there's even a mutated gamma secretase. When they prune the APP, you start getting these long strands. It can go up to 48. And then the body's like, wait, this is like really like long. It's getting kind of tangled. And then what happens is that it interacts with that apolipoprotein E. And if there's inflammation present, then it will start getting tangled on itself and forming these plaques. So you can see here, APP, amyloid precursor protein, is a glycoprotein. That means sugar plus protein. And here we have the alpha secretase. That's cutting it. Supposed to cut it right there. Great, cut it beautiful. Here's the gamma secretase. Cut it beautiful. Non-amyloidogenic pathway. That means you don't get the amyloid plaques, right? This is normal. Then it can do its job. Uh-oh. Here we go. Now this time we're getting beta secretase coming in. Oh dear. It gives it a cut. Still got the gamma secretase, and we got the amyloidogenic pathway because we got the long peptide chain. The body's like, does not compute. Not awesome. I don't know what to deal with that. Too long. So why? Why does this APP, amyloid precursor protein, not function properly? Beta secretase, we talked about that. It gets activated. We'll talk about later why it's activated, or there could be mutation of gamma. That's minor, more minor than the beta primary. Low estrogen, that plays into it. Alzheimer's is more common in women who are postmenopausal. Men have low estrogen also, but their receptors are not used to having higher estrogen. And improper exposure to metals. That's another reason that you can have improper function of the amyloid precursor protein. So this estrogen. So estrogen binds to the receptor and activates the alpha secretase. If you don't have as much estrogen as you're used to having, then you're going to have impairment of cleaving this amyloid precursor protein. And what we also know in mice, at least, is that estrogen downregulates beta. So it's kind of like a double whammy. If you have low estrogen, then you're not going to get activation of the alpha, and then you will be more likely to get activation of the beta secretase. And then the metal exposure, we know that the amyloid precursor protein responds to iron, copper, zinc, and that they need to be in the right ratios in order for it to function right. So that's a component. We also know that copper is found in the amyloid plaques. And of course, excess copper will cause death of the synapse and the neurons. Of interest is that when you get this long string of peptides, the long amyloid beta string, what happens is that it turns on or induces the hyperphosphorylation of tau. And that is the thing that causes the tangles. And why is hyperphosphorylation turned on? It's a normal part of development, and the enzyme that breaks it down, phosphorylase, is decreased with hibernation and hypothermia. So phosphorylation is important for cell growth and renewal. And so when you need that, you will have more of it. So here's the healthy microtubule right here. That's stabilized with the tau protein. That's part of the neuron axon. And then with the amyloid beta influence, then what happens is that you start getting breakdown of this microtubule and tangling up. And the tau is really important. It's supposed to, it's what stabilizes that tubule so that it can carry nutrients that keep the brain healthy. It's helpful for cell signaling, for synapse plasticity, so responsivity to the environment, and for the genomic expression. So it has a lot of different functions. And when it malfunctions, then it's just like a clump. It doesn't work anymore. It can't help the tubule stay open, can't carry nutrients, and then everything falls apart. So we're talking about misfolding of proteins. We're talking about problems when proteins misfold. So that's one of the things, right? protein misfolding in itself is a problem because the form of the protein is changed, so it can't do its job. And so we talked about amyloid beta and we talked about tau and how they're compromised in the ability to do their job when the folding is wrong. Other things that you probably are familiar with is like hemoglobin. That's supposed to carry oxygen. If hemoglobin is misfolded, like say sickle cell anemia, right, it can't carry the oxygen. So sickle cell anemia is awesome for preventing malaria, but not awesome for carrying oxygen. And so there's like this trade-off that's happened with sickle cell anemia, for instance. And if a mitochondria protein is misfolded, like we were talking about, you can't make ATP, so you don't have energy. So you can't like renew. You can't have energy. You can't continue living, basically. So that's one of the problems. We talked about misfolding of protein itself. The other problem with misfolded proteins is if they don't get cleaned up properly. So if you have a misfolded protein, at least it should get cleaned up, and then it'll be less of a problem. But if it doesn't get cleaned up, you have two problems on your hands. So the misfolding of proteins itself is caused by advanced glycation end products. That means a sugar and a protein combine, or a sugar and a fat combine. What do you get? You get trouble. So the thing that everybody knows about the most is like brown spots. Like I have a lot on my hand. These are all advanced glycation end products. This is all a product of sugar and protein, or sugar and fat combining incorrectly in a way that's harmful. So everybody knows about hemoglobin A1C. Hemoglobin A1C is something that we use to measure more or less how your blood sugar was over the past 3 months. But it's actually also measuring inflammation in your system, because it too is representative of being an advanced glycation end product. We talked about free radicals. When there's an uneven number of electrons in a molecule or an atom, that can cause misfolding of proteins. We talked about reactive oxygen species, which is a free radical in an oxygen environment that can cause misfolding of proteins. So the misfolding of proteins caused by the advanced glycation end products is a glycotoxin. That's bad. And if it combines with protein, you're going to get inflammation, you're going to get antibodies, blood vessel damage. It can be related to type 2 diabetes and type 1 diabetes. And it's when its receptor is involved, then you're going to get inflammation there in the receptor. And it can also produce its own free radicals. And then that goes on to just wreak havoc everywhere, even in the DNA and the cell membrane. So it's not awesome. So this is a picture of a free radical. So you can see it's like, I need an electron. Give me an electron. Best place to get it is with an antioxidant. So that's why you need to eat a lot of fruits and vegetables, right? Because they're super high in antioxidants, for instance. So stealing an electron from an antioxidant is the best thing that a free radical can do. That's the best place to get it. Now, here's the reactive oxygen species. These are free radicals in the presence of oxygen. And they're also like the hydrogen peroxide is one that you'll see in all of the like pathways that's thrown off a lot. And that's a poison. That's a poison. So what's supposed to happen to these misfolded proteins? They're supposed to be degraded. There's different pathways. They can be degraded along. The ubiquitin and the autophagy pathways are the ones that they're degraded on. So here's the ubiquitin one. So what impacts this pathway are things like epigenetics. metabolism. These things can inhibit the ubiquitin pathway if they're not done properly. And so what we need to do is to optimize the epigenetics, optimize our exposure, optimize our metabolism, optimize our stress level and how it's expressed in the endothelium or the this is what we're shooting for. So then when we talk about autophagy, we're talking about part of it is the chaperone. So you can see there's chaperone mediated autophagy here. That's what we're going to talk about next. This is chaperones. I don't like that name. I think they should be called nannies because they're not just like a chaperone. Let's say a chaperone is like your little brother or sister being sent on a date with you so no funny business happens. That's a chaperone, right? Nannies do a lot more than that. And so these chaperones are more like nannies. They're controlling the quality of the protein because what they do is they oversee the folding of proteins. Like a nanny, let's say the nanny is helping you clean your room. The nanny will be like, okay, this is how you fold your shirt. Now you give it a try. So make sure that you fold your shirt the right way. And then the nanny would be like, pretty close. Let me show you again. And then would fold it correctly. That's a nanny does that. Chaperone doesn't do that. That's the way I'm thinking about it. And then after the nanny oversees cleaning the room, then the nanny will be like, okay, now let's take all the dirty clothes out. That's kind of chaperoning piece of it. But also that's what a nanny does. So that's a normal part of cleaning up misfolded proteins. So one of the chaperones, I call them nannies, is a heat shock protein. These are awesome heat shock proteins. They help fold new proteins. They help prevent the proteins from sticking together. They are good at breaking down misfolded proteins. They transport the proteins, and what's really special about them is that really good at finding the beta amyloid plaque. When they find the beta amyloid plaque, they're like 911. They attach to it, and then the glial cells are able to like see it. They're able to identify it and then attack it themselves. These are the glial cells. Glial cells are in the brain. So we talked about the microglia before. The astrocytes are glial cells that are for the blood vessels, and the oligodendrites are the glial cells that are for the myelin part of the neuron. Those are the glial cells. And so the microglia, they're supposed to help clean up inflammation. So if there's trauma, if there's infection, anything that creates inflammation, they'll be like, let me handle this. And so there's a few different kinds of microglia. And what happens with initial exposure of a pathogen or a tumor or something like that is that there's a pro-inflammatory response. This is normal because they're trying to kill it. So that's normal. So that should be turned on and turned off. And then also we have the M2 or microglia number 2, which is an anti-inflammatory microglia. This is the one that promotes tissue remodeling, repair, angiogenesis. And so with acute disease, this anti-inflammatory M2 microglia is neuroprotective. That's awesome. But what happens is that with chronic exposures, chronic disease, the brain gets switched over into this neurotoxic M1 dominance, which is pro-inflammatory. And so like the cells that are supposed to be cleaning up the mess actually become part of the mess. And so that's why we want to intervene as soon as possible. We don't want to let things, spiral out of control, because the body becomes dysregulated in its pro and anti-inflammatory systems. And so here's an example of Alzheimer's. So this is your microglia, it's resting like tra-la-la. And then we have early Alzheimer's. And what's supposed to happen is that most of that amyloid beta is going to be cleaned up, discarded, pooped and peed, sweated out. Some of it might not be, and then that's going to be shifted up into a more chronic state. The microglia, then in the advanced or chronic Alzheimer's disease, we have the activated microglia. And then we're getting, remember those RAGE, the receptor for the advanced glycosylated end products? Ooh, those are here. We have inflammation already. Ooh, this is the beta secretase. Ooh, that's coming on. Oh, and now we're getting all these pro-inflammatory cytokines going on. And now we've got the amyloid beta accumulation and neuronal damage. So that's what happens when we have chronic activation of microglia. They become pro-inflammatory. So again, these heat shock proteins, that's where we started at. They're the chaperones. They're the ones that help us clean up things, help to identify the beta amyloid plaques so that the glial cells can like attack it, try to clean it up. They're super awesome and important. And so we need to upregulate them. So the main way that they're upregulated is they're called heat shock proteins. It's through heat. So taking a sauna is really awesome. Trying to sweat every day, if you can, is awesome. A lot of people have a problem sweating. It's hard for their body to sweat. And so if you can get in the habit of sweating, this is awesome. Definitely do not use antiperspirant because you need to sweat. So it's funny though, because these heat shock factors that activate the heat shock proteins are not only hypothermia. Look here. There's hypothermia, hyperthermia, too much heat, sauna. It's also hypothermia. So that's like cryotherapy. So now cryotherapy is like a thing for chronic disease. You have to go into like this really super cold place because that's another way that the heat shock proteins get turned on. This is a combination of ischemia and reperfusion. And so nowadays there's a kind of therapy where you tie this like kind of like a rubber band around an area of your body to create ischemia, which is where there's no blood flow, right? And then you release it, and then there's hyperperfusion. And so that's another way to turn on the heat shock proteins. In manual therapy, we use our hand to do something that's called a blood stop, where you would use your hand to press down on a vessel, create a blood stop, and then release it suddenly so that you can get that hyperperfusion, turning on the heat shock proteins. And it's funny, right? Because both, again, hypoxia and hyperoxia, so not enough oxygen, too much oxygen, can also turn on the heat shock proteins. So they can be like the Wim Hof breathing, for instance, the oxygen tanks, for instance, the highly pressurized oxygen, and energy depletion turns it off. So basically, like, you know, when the body's in a extreme state, this is going to turn on the heat shock protein. So this would be like fasting. Acidosis, this would be like ketosis, because you put you in a pH that's acidotic. Viral infection, the body's like, got to get to work here, turn on the heat shock proteins. The reactive oxygen and nitrogen species will do it too. So one of the main reasons that misfolded proteins are not cleaned up is because the insulin degrading enzyme is used both to clean up the amyloid beta plaque and insulin. So if there's too much insulin circulating in the brain, the insulin degrading enzyme is going to be like, I'm going to be working on that insulin instead of the amyloid beta plaques, because working on the insulin, degrading the insulin is my number one priority. Insulin is highly inflammatory. You think glucose, you think sugar is inflammatory. Insulin is also inflammatory. So the body's like, I don't want that inflammation. I'm going to degrade that. And then I'll work on the amyloid beta plaques when I have time. So we have this shift away from addressing the amyloid beta plaques to addressing the insulin by the insulin degrading enzyme. So when we think about insulin, we think, oh, that's like that. That's what escorts the sugar into the cell. So you actually have energy. It creates the glucose homeostasis. But insulin does a lot more than that. Insulin and the receptors in the brain are also important for neuronal integrity. They help us think, cogitate. They help with the neurotransmitters. And again, we have the NMDA receptors that are for energy, for balance, and anti-inflammatory when done right. The insulin receptors are important for maintaining the excitatory synapses. So that's the glutamate, that's the acetylcholine. They help with the insulin receptors help with the dendritic spine formation, and also, again, the apoptotic pathway. So breaking down, taking away of cellular debris. So this is the reason that Alzheimer's is called type 3 diabetes. Because a lot of people who have Alzheimer's have diabetes of the brain. They have too much sugar in their brain, too much insulin in their brain. So the insulin degrading enzyme cannot work on the amyloid beta plaques. They're working on the insulin. So what you see with Alzheimer's is progressive insulin resistance in the brain. This leads to neurotoxicity, neuronal stress, neuronal degeneration. Because you have too much insulin, you're not getting the amyloid beta plaques cleaned up, and and you're also getting impaired insulin signaling. So it's a problem upon a problem upon a problem. Too much insulin makes amyloid beta accumulation inside of the neurons. And then you have that breakdown into tau, and it's this slippery slope. Everything is falling apart. It's becoming very sticky, very inflamed. So amyloidosis, or the amyloid beta plaques, are not the cause of Alzheimer's. And that's why all the research has failed. That's why the drugs fail, because it's not the cause, but it's a mechanism of pathology. Something else is driving it. And that thing that's driving the amyloidosis is the thing that we should be looking at, not the amyloidosis itself. So Alzheimer's is a synaptic failure that is caused by the amyloid beta plaques and the tau hyperphosphorylation. But something is driving that. And that is what we need to look at. That's what we need to find. And that's what we need to address. And that's what this program is about. And so even though, we know a lot of things like the shrinking of the hippocampus, we know there's a new protein that they've discovered that's related to Alzheimer's disease. It's not the cause. It's related. We'll find out more about it with time. We know that the mechanisms have to do with misfolding, mitochondrial dysfunction. Those are not the cause. That's part of the problem. It's not the cause. And the problem is with all the research is that we're not looking at the right thing as the etiology, as what's turning on this process. And we want to change that. So for Alzheimer's, there's a progression where you're losing smell, you're losing your ability to identify flavors, you're losing your episodic memory, which is the hippocampal memory, you're losing cognition, there's emotional changes that happen, aggression, losing the ability to speak and communicate properly, find yourself in time, day, night, all of that, confusion, leaving, getting lost, not knowing where you are. Is it day? Is it night? I don't know. And then losing your ability to move properly and swallow and to recognize loved ones. And this is the progression that breaks your heart, that we want to avoid. We want to step in here. We want to step in here when we first start noticing, oh, something's happened in here. We want to address that. That's what this program is about. Biomedicine, want everything's normal until you can't recall somebody's name. That's too late. So our 3 phenotypes: inflammatory, the predominant one; atrophic, not enough nutrition; toxic, something's there that's agitating, that the body's fighting against or trying to seal off. We have to get rid of. We talked about the methylation. We need to be able to methylate properly in order for everything to heal. We have to be able to produce and transmit neurotransmitters. We have to have a well-functioning immune system, vascular system that has proper flow. We have to be able to produce and use energy and have a strong barrier system. Those are all the things that we need. And we're going to take a break before we start talking about all those different systems. So now we're talking about the systems that impact cognitive decline. Methylation. So methylation is one of the processes in our body that basically just means you're adding a methyl group to something. And so this is required for DNA. And part of it, one of the things that's been pretty highly studied is homocysteine, where we know that if we get a high homocysteine, we're going to have brain atrophy. It's very inflammatory. If we get a high homocysteine, it's going to be very hard on the heart. And so homocysteine is a demethylized product. Basically, the opposite way of saying that is that methionine is methylated homocysteine. And I'll show you in a second the chart so you can understand that a little bit better. So methylation is not only like adding a methyl group, it needs it needs helpers. And when it has these helpers to get the methyl group attached to something, then what we're going to do is be making vitamins. We make amino acids, we make neurotransmitters, we make hormones, red blood cells, DNA, RNA, antioxidants. Methylation is super important for loads of activities in the body. So we talked about the neurotransmitters. So melatonin is a byproduct of serotonin, glutathione, part of the transsulfuration pathway, coenzyme Q10, important for heart, some amino acids, and then phosphatidylcholine, which is part of the methylation pathway. The choline is, and then choline, acetylcholine, the neurotransmitter, has that, and then also related to making the myelination, the myelin sheath around the nerves. So the thing that helps to methylate, which is to say, to donate the methyl group to another substrate, are the B vitamins, betaine, choline itself, and then SAMe. These are the things. Now, everybody nowadays knows about the different SNPs. So single nucleotide polymorphisms, that's a mutation or variant, genetic mutation or variant. And so the one that people talk about the most probably is the MTHFR. So the MTHFR mutation 677 can impact homocysteine production. So that if you have heterozygous, so that means you have one variant and one normal from your mom and your dad, then you'll have 40% reduced activity. And you're gonna, if you have homozygous, which means that you have a variant from your mom and your dad that is not, well, it's a variant, right? So it's a mutation. So this will reduce it will reduce the methylation of homocysteine by 70%. And so you'll have a very high homocysteine level. I'm going to show you the picture. It's going to make much more sense. So here's the MTHFR. This is the folate cycle right here. This is the methylation cycle. Here's homocysteine. Homocysteine is when it becomes methylated, that's when it's methionine. It switches its name into methionine. Methionine is a methylated homocysteine. So if you have a MTHFR 677 SNP, then it's going to slow down this process right in here. This is the B12 or cobalamin process. So it can have its own SNPs that slow down, speed up. And so homocysteine will have a hard time getting up here and becoming methylated to become methionine if there's an MTHFR SNP. Homocysteine can take this other route up this way. If there's a BHMT SNP, then this also can be slowed down, or with the MTHFR, MTRR SNPs, it can just run this pathway too much, so it can't keep up with it. And so anyways, you're going to still end up with a high homocysteine. And that's going to lead to brain atrophy and to cardiac problems. That's how methylation plays into it in part. High homocysteine atrophies the brain. Not good. Now we're going to talk about the neurotransmitters. So the neurotransmitters are the acetylcholine, that's excitatory, GABA, inhibitory, serotonin, inhibitory, dopamine, catecholamines. Glutamate's not on here, but we've kind of been talking about that. It's also excitatory. So acetylcholine is like the main one that's associated with Alzheimer's and then also cognitive decline. And especially the part of the brain that's called the hippocampus. And we already talked about the hippocampus, because in the hippocampus, the dentate gyrus, that is where we get that new neuroneogenesis, right? That's where new neurons can form. So if we have low acetylcholine, the symptoms that we're going to have are a poor memory, slow mentation, difficulty finding your way around, losing your keys, losing your phone, your car, your glasses, can't keep track of your stuff, need to use your GPS to get around, have to write down stuff, can't remember it, can't get to your appointments, and you'll get into a room and wonder why you're there. You know you went in there for something, but what was it? This is not good. These are things we accept as normal, and they're not normal. They're not good. It's brain degeneration. So acetylcholine is produced in the axon of the nerve. So it's produced right in here. And so what it is, see, this is a mitochondria again. Mitochondria is part of the Krebs cycle. And here's choline. Choline is part of the methylation cycle. So these two are going to hook up. Acetyl-CoA, acetyl group hooks up with choline to make acetylcholine, the neurotransmitter. That's how it happens. You get acetyl from sugar plus the choline, acetylcholine. Neuroexcitatory, and at the same time, anti-inflammatory, improves your memory. So the main place where we're interested in Alzheimer's, where you know this is the number one first place that is affected, is the basal forebrain, and this is what has the projections to the hippocampus for memory. So this is a really, really important area. So right here, see, it says hippocampus. That's where the hippocampus is. The hippocampus, like I said, in real life. Looks like a seahorse. Doesn't really look like that here. But you can see, here's the amygdala, part of the limbic system. It's also part of the memory system, converting short-term memory to long-term memory. On the left side, it helps with vocabulary, finding the right words. The right side, it helps with spatial orientation. And so what's supposed to happen is that there's supposed to be like a turning off and turning on. In order to make new memories, we have to allow for old memories to become more flexible so that we're not stuck in the mud. And that's part of what the hippocampus does. That's what part of what acetylcholine does. This Papet circuit is what helps us with the emotional or limbic piece of the hippocampus and acetylcholine. And what's supposed to happen is that we're supposed to be able to organize our emotions instead of becoming overwhelmed by them. We're supposed to be able to take in new information and remember it and use it and then convert it to long-term memory. And so if we have a problem in the hippocampus, if we have shrinkage, if we don't have enough acetylcholine, all these things become difficult. So here's choline. So we were talking about that before. Acetylcholine is a neurotransmitter. That's, combination of choline with the acetyl group here, the Krebs cycle. Choline doesn't only go into being a neurotransmitter, it also goes into helping create the myelin sheath. It also goes into the methylation cycle, working with that short track of methylation, bringing homocysteine up into methionine, the shortcut. That's where choline works. Right. And so this is what we were talking about before, is that one of the jobs of acetylcholine is to suppress the old associations. So again, this is inhibition. Inhibition is very, very important for high function. So we have to inhibit in order to create something that's new, in order to take in new information and make meaning with it. Acetylcholine also works on the autonomic nervous system. So the autonomic nervous system we talked about before with babies and how they go from being sympathetic to parasympathetic dominant as they grow up. And anyways, that's what's supposed to happen. So all of the preganglionic neurons are run by the acetylcholine, both sympathetic and parasympathetic. So those are the preganglionic parts of the autonomic nervous system are in the spinal cord, basically, brainstem. And then the postganglionic neurons, the acetylcholine's action is all along the parasympathetic side and also the sweat from the sympathetic. And so you can see here, ACH is acetylcholine. Acetylcholine is very, very impactful for the parasympathetic nervous system, all these things. And along with the vagus nerve, so we have vagus nerve, we have acetylcholine that have bidirectional anti-inflammatory communication between the brain and the periphery. So the vagus nerve we saw before as being a major inhibitory factor for the sympathetics. And acetylcholine works primarily on the parasympathetics. And so these two things are very, very important for the anti-inflammatory path between the brain and the body. GABA. GABA is the next neurotransmitter. It's inhibitory in nature. So when there's not enough GABA, then a person's going to feel anxiety, panic, they can't turn off their brain, they're ruminating, they feel overwhelmed, and they're like, but I used to be able to manage this. Now it's like too much for me. I can't do it. And I can't really start anything because I see this needs to be done and that, and I don't know where to start. There's just too many things. So that's like a low GABA person. These are the people who, if there's something happens, they're going to startle. And they're also like very sensitive to light and sound when you're low GABA, because it's inhibitory. You're all like way up into glutamate, the excitatory the excitatory neurotransmitter. So of course, GABA cannot cross the blood-brain barrier because the neurotransmitters are too big. They can't cross the blood-brain barrier. They're made inside the brain or outside of the brain. They can't cross it. And GABA, in order to be synthesized, it requires the B6. Well, this is where it goes. It goes everywhere in the brain, basically. But I want to show you right here. This is what I want to show you. So glutamate is the excitatory neurotransmitter that breaks down into GABA. But it can't do that without B6. It needs B6 in order to do that. And in order to have B6, we need to have proper methylation. This is a gene. A gene is an enzyme. This is glutamate decarboxylase, which is the GAD gene. The GAD gene. The most common GAD gene to have a genetic SNP in is the GAD65. And that one is related to the generalized anxiety disorder. So this, when you can't break down glutamate into GABA, you get anxiety. And what you need is B6. What else do you need? What else works on here is gluten. Gluten will block this enzyme, so you can't break down glutamate into GABA, and you'll end up with excessive glutamate. So that's one of the reasons that people have low GABA, is because they're not actually converting glutamate into GABA. They're ending up anxious, overwhelmed. So these are more genetic SNPs. So we talked about MTHFR 677 and how it affects, how SNPs or mutations affect the methylation process. We also have a different form of MTHFR 1298 that can impact the biopterin pathway, which is the pathway that creates serotonin, dopamine, norepinephrine, and epinephrine. And if you have a genetic SNP for this one, then you're going to have a problem creating those neurotransmitters, and you're also going to have a problem detoxing ammonia by the biopterin pathway, because the biopterin pathway is just going to be slowed down. Ammonia is neurotoxic. The brain cells die when exposed to ammonia. It's not good. So this is this from the folate pathway, right? And how that can impact the neurotransmitters, brain health, in a variety of ways. COMT is another gene, which is an enzyme, which helps to break down dopamine, and it can either speed up the breakdown of dopamine or slow it down. Same with MAO. MAO-A is the one that's most significant. It's for serotonin and also dopamine, and it can either speed it up or slow it down. And so these are genetic SNPs that are impactful on the neurotransmitters. So again, here's the MTHFR. So we say the forward swing, the 677, impacts methylation and creates that high homocysteine. That creates brain atrophy and cardiac problems. The backward swing is what goes into the MTHFR. That's what goes into the biopterin pathway for serotonin, dopamine, norepinephrine, and epinephrine. And that's the one, when you have a genetic SNP here with the backward swing, the 1298, then what you have is a slowdown of the biopterin pathway by and large, and a difficulty clearing out ammonia from the brain. And then that is neurotoxic. So this is the biopterin pathway right here. So you can see that this is, these are the precursors for dopamine and serotonin. So we need to have the amino acids, the iron, the B's, and then when we make dopamine, it's not enough to just have dopamine. We should have it, and then it should get broken down. If things stay around too long, then it also is not good. It's not having proper function. So dopamine, if it's broken down properly by Compton Mao, others too, but primarily, then if it's made properly, broken down properly, awesome. If it's not, then we can end up not only with high dopamine, we can end up with high norepinephrine and high epinephrine, which is adrenaline. That doesn't feel good. And then here's serotonin. Serotonin also requires the amino acids, iron, B, to be broken down into melatonin, and then its byproducts by Mao and others. So serotonin is produced in the brain and in the gut. If you have low serotonin, then you're like, why do I feel bad? I should feel better. I have a great life. Nothing's wrong with my life, but I just don't feel happy. That's serotonin. And it's the one that's associated with seasonal affective disorder, because that's the raphe in the is what kind of has receptors for light. And so when there's not enough light, then you're not able to make the serotonin in the brain. And so you get the seasonal affective disorder. And because it breaks down into melatonin is what comes up to make us sleepy and then comes down to make us wake up. So if you're not getting enough serotonin, you're not getting enough melatonin, so you have disrupted sleep cycles. And so the body's like, let me help you out. Let me give you more carbohydrates so that you can try to produce more serotonin. And people just feel like, oh, I hate everything. I just want to be by myself. I don't feel like doing anything. So that's serotonin. Here it is. Here's a raphe nuclei where serotonin is produced in the brain. Then it, of course, goes everywhere in the brain. But only 5% of the production of serotonin is in the brain. The rest of it's in the gut. 95% is in the gut. So what we know now, a lot of affective disorders, a lot of the signals, the signs that go along with cognitive decline are actually gut issues. And we've talked about autonomics, but also the neurotransmitters are related to that. And so serotonin is one of the primary regulators of the enteric nervous system in the belly. It's responsible for motility and propulsion. I mean, parasympathetics do that too, but they both play in the same way. It regulates the immune system. And then talk about leaky brain, leaky gut. Serotonin is what regulates the epithelial integrity of the gut. And so it's responsible in part, for keeping that sealed so you don't get the leaky gut. So we talked about serotonin production here in the biopterin pathway and how we need to have that good folate to feed into it, that good methylation to feed into it. Now, dopamine. When you have low dopamine, this is a person who's unmotivated. Very hard to start tasks because they just don't feel like it. Once they start it, that makes them feel better. So they can go for a little while because doing a task that you like it creates dopamine. But then usually they're going to peter out. They can't finish their task because they get distracted by something else that needs to be done. So they'll start and not finish. And there's like really so sad. They beat themselves up. And they rely on caffeine and sugar to be able to try to get a dopamine high, because those things do help to create dopamine. Alcohol, gambling, those things also help create dopamine. So these folks also are going to isolate, but then they fly off the handle. And that anger creates dopamine. And these are like, my teachers always said, the dopamine patient is the hardest patient to deal with because they have a real hard time doing anything. They don't have compliance. Maybe they're going to do their exercise once, but they take their pill here and there, but it's very hard to regulate for the dopamine, the person who's low in dopamine, and they're constipated. So now, Parkinson's is related to dopamine, low dopamine. So now the idea is that Parkinson's actually starts in the belly and that it works its way up. It crawls up the vagus nerve into the brain. And that the first symptom of Parkinson's is actually constipation, many decades before you see Parkinson's symptoms expressed. So here's the dopamine pathway created in substantia nigra, ventral tegmental area, and the hypothalamus. Again, none of these neurotransmitters cross the blood-brain barrier, all created in situ. Again, here's the dopamine pathway. We have to break it down. High dopamine is going to create anxiety, aggression. Catecholamine neurotransmitters. These are dopamine, norepinephrine, and epinephrine. So the breakdown products of dopamine and dopamine itself. When you have low catecholamines, you're going to have slow cognitive processing. Again, easy distractibility. Again, the reliance on caffeine. Everything is slower. Use the caffeine to try to get that high. We see again the dopamine here breaking down into the norepinephrine. That creates alertness if you have adequate dopamine, then you're like, I want to do that task. That sounds great. Let's go do it. And then that'll break down into norepinephrine. And you're like, I want to keep doing that task. Let's finish it up. And then if you have, where's my pointer? If you have adequate breakdown here with PNMT, then you're going to get the epinephrine, which is the adrenaline, and you'll be like, I feel awesome. I did this great task. I started it, I did it, I finished it, now we're done. Let's have a party. So that's the way things are supposed to work if there's no SNPs and if there's proper epigenetics, nutrition, so forth. So those are the neurotransmitters. Now we're going to go on to more systems that impact brain health. We talked about methylation, neurotransmitters. Now we're going to go on and talk about the inflammatory and infectious cycles. And so we've kind of talked about this before when we were talking about breaking down the misfolded proteins and stuff. So inflammation is what is supposed to happen in the body when we have an infection, when we have trauma, pathogens, or irritants. It's the normal and right response to have. The problem is if that inflammatory pathway does not get turned off. If it does not get turned off, either because of confusion in the pathway, chronicity, or if that pathogen or irritant, that trauma doesn't go away. And so then it stays turned on trying to battle that until it runs out of steam. So this chronic inflammation leads to cognitive decline. Number one, in Dr. Peterson's modeling. So interleukins, IL is interleukin. So you're going to see that over here. Interleukins can be either pro-inflammatory or anti-inflammatory. They are cytokines. Cytokines are secreted in the nerves to regulate immune response. Cytokines. And so what we need is a balance of pro-inflammatory and anti-inflammatory. And so we need the pro-inflammatory initially, then turn off. Then we need anti-inflammatory for rebuilding. And what we need is the pro-neurogenic lifestyle in order to make this happen. And that's what we'll be talking about going forward. The vascular system. So vascular dementia is the second most common form of dementia in the United States. But different things can impair the vascular system and its ability to provide nutrition to the brain. So if you have a low flow, low perfusion, you're going to have that hypoxia and ischemia. Remember, the body's going to respond. It's going to be like, oh, 911, get the heat shock proteins going. So the body's going to like try to get things cleaned up, but it may not be able to. So nitric oxide, the endothelial nitric oxide is supposed to create nice open blood vessels so we get good flow. But there's also a couple other varieties that can create inflammation. Another problem that people have is not with low pressure, but with high pressure. And that's when you're going to get the ruptures, the aneurysms, the strokes. The stroke increases the probability of the vascular dementia. And we know inflammation also is associated with poor blood flow, and that's associated with Alzheimer's too. So now we want to talk about how the body has to be able to produce energy, right? And so the aerobic respiration, so using oxygen, we produce ATP in the mitochondria. We need carbohydrates, fat, protein, B vitamins in order to produce that energy. And then the body has to be able to use it. So the energy has to go to the right place. The cell has to be able to take it in. And we know that the brain is very demanding. Even though it's a tiny bit of mass, it uses a huge part of the body's energy. And most of that is used by the neurons in the brain. So what are the things that make it hard for energy to get into a cell? So we have to make the energy, and then we have to, the energy is a fuel, right? Then we have to actually be able to use the fuel. So the fuels for the brain are primarily glucose, oxygen, fat, and protein. So we need, if we have insulin resistance, then what's going to happen is that the fuel, the glucose, is not going to get into the cell. It's just going to be circulating like on the outside of the cell. And so we don't have fuel, and we do have inflammation. And then we have high insulin, and then our insulin degradation enzyme is going to be working on that instead of on the amyloid beta plaques. And so that's a slippery slope. Oxygen, we need the oxygen. And the oxygen, there's no reserve of oxygen. You have to breathe it in all the time. So that means that you have to have proper vascularization in order to have appropriate perfusion of the oxygen into the cells. And then we know we have to have adequate fat for the nerve transmission. Protein is what the neurotransmitters are made out of. We need all of those things for fuel for the brain. Have lack of fuel. If we have blood sugar dysregulation, it could be high or low. If we have anemia, so we talked about the sickle cell anemia, misfolding of the hemoglobin before. We could also have anemia that's because we don't have enough B vitamins or enough iron, the cofactors for making the neurotransmitters. We can also have a lack of fuel. Well, like we talked before, you don't get oxygen, you got poor perfusion, low nitric oxide for dilating the blood vessels. Hypothyroidism can also do this. And then diet and exercise feed into this, because remember that the mitochondria live in the cells. And if we don't get exercise, the mitochondria are going to die because there's no point. Body's very efficient. So in our model that we're working with, most cognitive decline is related to the inability of fuel to get into the cell. The second most common reason is because there's not enough fuel. We don't have enough nutrition. It's not being carried properly, or the hormones. The last one is toxins. And basically what toxins do is they steal fuel, they steal energy, and use it for other reasons rather than for maintaining the brain. So in the hypertrophic model, the low thyroid function definitely can contribute to this. The thyroid hormones are really important for maintaining hippocampal volume. And remember, that's where the dentate gyrus is, where new neurons are formed. The thyroid hormones help with all the sort of like processes and stages of neuron development. So if we don't have enough thyroid hormone, we're going to have downregulation of the brain. And if we have too much thyroid hormone, it can do similar things where it creates a mad brain. It creates craziness in the brain. So we don't want too low. We don't want too high. We want just right. Goldilocks, Goldilocks. And then the last system is the barrier system. So we talked about the blood-brain barrier. You have the 1 gram of GABA that you're going to use to see how your blood-brain barrier is doing. We'll talk about that next time. And it's the phospholipids that are primarily what form the barrier system. And the phospholipids is basically fat. Choline also helps with that. This is what creates safety between the inside and the outside of the brain. Now, normal aging, you don't really see a change in these phospholipids. Normal aging, the brains of a 40-year-old and a 100-year-old are just about the same. You can maintain a strong barrier system. We're designed to. We need that. But with abnormal aging, cognitive decline, you're going to see a loss in that barrier system. And you're going to start getting infiltrate from the outside of the brain into the brain. And that's why they say with Alzheimer's, it's like a zoo in there. There's so many microorganisms because they have a leaky brain. And then, of course, we're going to have loss of the gray matter and white matter in Alzheimer's. And so this is the progression that is abnormal aging and something that can be avoided in many cases. We need those phospholipids. We need cholesterol homeostasis, triglyceride, which is a storage form of carbohydrates, and then the membranes that separate and keep things safe. Now, remember, all of this, everything that we're doing is based on the 3 phenotypes of cognitive decline that Peterson uses: inflammatory, atrophic, and toxic. Those are the things that we're going to go into more next time. Inflammatory phenotype, we've talked a lot about high blood sugar and insulin, major obesity, being overweight even contributes to that inflammatory cycle. Exposure to foods that you're intolerant of, too much stress, that creates inflammation. Incorrect ratio of zinc to copper, also inflammatory. Low glutathione and selenium, inflammatory. Sleep apnea, inflammatory. Leaky brain, leaky gut, leaky, inflammation. And then the chronic inflammatory response syndrome, CIRS, which is another thing that we'll look at. And then autoantibodies of the central nervous system. These are all components of that inflammatory phenotype that have to be teased out and find out what is, if any of them are relevant for you, and then addressing them. The atrophic phenotype is the one where you don't have the nourishment that it takes to make proper brain cells and to have proper communication. So this can be because you're not getting enough cholesterol, either because you're not making it or because you're taking a statin to bring it down low. It could be because your steroidal hormones are low because you're in menopause or andropause. You could have a low thyroid function, low magnesium, again, sleep apnea. The fat soluble vitamins, B1, and then mitochondrial damage. All these things make it hard to have the nourishment, the energy that we need to have a healthy brain. And then the final one, the final of the three phenotypes, is the toxic phenotype that causes cognitive decline. This could be because of exposure to mold or mycotoxins, heavy metals, or a number of different infectious agents that undermine the quality of the brain. So again, our goals are to identify if the blood-brain barrier is sealed. If it is not, then we'll seal it up. Then we want to see what factors, what systems are involved for you in cognitive decline, identify what those are, and then make a treatment plan for that. If there's something like in the toxin range, in the infectious range, something like that, in the food range, then we want to take those things out and then bring in the pro-neurogenic lifestyle as we rebuild the neurons and the synapses in the brain and bring back full function. So this is a lot. I know this is a lot. And so ask yourself, take some time, ask yourself, how am I feeling about this information? What is it? What are my reflections about myself, my own physical and mental health? Am I thinking about somebody that I know and love? This is relevant for them. What am I feeling about that? Am I ready to start a program? Is this the right time of my life to bring in a program to improve my cognitive health? Do I have the time and the interest and the energy to do that? Is this the right time? If so, it's the right time. How do I want to start? Am I the kind of person who likes to dive into something and go whole hog? Or am I the kind of person who likes to do change incrementally? What works best for me? How can I make long-lasting change? What is the best way for me to make long-lasting change? These are the questions to ask yourself. If I am the kind of person who likes incremental change, then how do I know when it's time to step up to the next thing? How do I know when to augment the intervention? The other questions that are important to ask about the diagnostic testing, and especially about the Alzheimer's markers and genes that are related to Alzheimer's, like the APOE4, how do you feel about knowing that status? Is that something that would be empowering for you? Or is that something that's scary and that would make you feel resigned? You don't have to get these tests. How do you feel about not getting the diagnostic tests? How do you feel about just moving towards the pro-neurogenic lifestyle without really knowing exactly what's going on with your own, diagnostic workup? How do I feel about kind of being blind in a way, but making changes that I know generically are helpful? So those are things that would probably be helpful to think about this week. And I know it's a lot. Just take it a little bit at a time, and you don't have to know everything. You don't have to memorize everything. It's okay to go through this slowly and do what's right for you. And we'll meet you on the discussion board.